Hello, Dr. Sodana. Today I will be presenting on our semester-long project in our cell biology lab entitled Screening Novel Compounds for Anti-Cancer Activity. This research was performed by Group 4, which consists of myself, Daniel Robledo, Melvin Onya, and Tyrone Miner. I would like to start off by giving you an outline of what I will be talking about in this presentation. First, I will give you the introduction, which is going to be about normal cells and cancer cells and what is the difference between them. Then I will give you, I will state the goal of the project and what we're trying to accomplish in here and why are we doing this research and why is it so important. After that, I will go over the experiments and the data along with the results. And finally, I will go over the significance of our results and end the presentation with the conclusion to our entire research that we conducted for this semester. Before I explain what we did in our research, I have to talk about normal cells and cancer cells. An adult human contains trillions of cells, which are utilized to grow, develop, reproduce, repair damaged tissue. Those functions are possible because of cell division. Each normal cell has some type of specific function in the body, and when the cell grows old or becomes damaged, it undergoes through apoptosis, which simply means a programmed death. Apoptosis is a critical cellular procedure that helps the body keep a healthy balance amongst cell population. Eukaryotic cells that lose their capability to undergo apoptosis have no way in controlling the cell population. Cancer cells are cells that cannot stop dividing and end up spreading into surrounding tissues. An important difference between the normal and cancer cells is that normal cells have specialized functions, whereas cancer cells do not, which is one of the reasons why they continue to divide without stopping. According to Cancer.org, roughly around 585,000 people will die this year from cancer, and a little over 1 million people will be diagnosed with some type of cancer. There is no cure for cancer, only treatments, and often these treatments that are available have negative impacts on normal healthy cells. Chemotherapy is an example. It does have some success, but also that type of treatment cannot differentiate between healthy cells and cancer cells, which causes many negative side effects on the host that's undergoing through that therapy. So that's kind of the significance and importance of what this research is doing, which kind of ties into our objective, which is the overall objective of this project is to find novel compounds that show anti-cancer activity against the cancer cell line U937. The anti-cancer activity refers to a reduction in cell proliferation, inducing apoptosis, and or indications of apoptotic markers in cancer cell lines. Compounds that show positive anti-cancer activity can then be further studied in hopes that they may become an anti-cancer drug in the future. With that being said, the first assay we performed was a MTT assay. This is just the raw data that we got out of our experiment. The objective of the MTT assay is to find the optimal number of cells needed to perform the screening process for anti-cancer compound trials. The MTT assay is utilized to measure the cell proliferation rate in the cultures, specifically the ones that we're working with, U937. The MTT assay is a colorimetric assay which analyzes cell viability. Based on the absorbance that is shown, we ended up using 50,000 cells for the next MTT assay that would contain the novel compounds that are being trialed. How this works is that MTT assay has specifically a yellow tetrazolium, and in the presence of metabolically active cells, tetrazolium is reduced by dehydrogenases enzyme. The dehydrogenases enzyme is reducing the tetrazolium by means of oxidation reduction reaction. When the yellow tetrazolium is reduced by active cells, a purple formanza can be analyzed using spectrophotometric equipment such as a microplate reader. The following MTT assay contain the cancer cell line U937, the compounds listed. The assay was used for screening a variety of compounds to see if the compounds being tested had a direct effect on cell proliferation or some type of cytotoxicity effect on the cells, which is indicated by the graph. You can see the percentage cell survival, and each of these compounds, as listed underneath the bars, it shows you what kind of effect they had on the percentage cell survival. Unfortunately, we only had two compounds, colchicine and indubulin, that had a reduction of at least 30%. BA3 showed some signs of cell survival reduction, but there was a huge error bar that discredited its results, and all other compounds, TRP1 through 7, did little to no reduction in the cell survival. Since indubulin and colchicine showed positive results in stopping cell proliferation and increasing cell death, we went ahead and performed an IC50 on both of those compounds. This is the raw data for the first compound being trialed, which was indubulin. Based on these data, we were able to make a graph which did not show the results that we were looking for, unfortunately. The results were questionable for indubulin in regards to the concentration of the compound. The compound concentration of 0.02 to 2 micromolars had no effect on the cells. But as soon as the concentration goes up to 2 micromolars, the percentage cell death jumps all the way to 75%, as seen right here on the graph. And it stays the same throughout the concentration, even though the concentration goes up to 20 micromolars. With that data obtained from this part of the experiment, we just couldn't determine a true IC50 for this compound. These are the results for colchicine in regards to the IC50 when we're trying to determine the IC50 for this specific compound. The results for colchicine were in fact much better than indubulin. As you can see on the graph, we can actually determine an IC50, which just by looking at the graph, you can kind of guess that it happened somewhere along before 1 micromolar of the concentration of the compound. But after calculations, we determined that IC50 for indubulin was around 0.025 micromolars. So after determining the IC50 for the compounds, then we proceeded to the next assay, which was the caspase assay. This is just showing you the raw data that we accumulated for the caspase assay, and using these data points, we were able to make a graph which was a lot easier to analyze. But basically, the first objective of this experiment is to determine if the apoptotic marker caspase 3 and 7 is present in the cell culture containing the novel compounds colchicine and indubulin. What this is basically the presence of caspase activity is a clear indication of apoptosis. Caspase activated DNase, or CAD, is actually a protein that breaks down DNA into fragments and induces apoptosis. Caspase 3 and 7 actually cleave off the inhibitory CAD protein, which once it gets cleaved off, it actually activates CAD. The presence of caspase activity can be analyzed by fluorescence. Based on our results, we can see that samples containing indubulin had the most caspase activity, which is this purple linear line right here. And it's actually followed by colchicine, which is the green line right underneath it. So those were the two compounds that we were testing for to see if it had positive caspase activity. And we tried this all the way to 150 minutes, and this is the results we got. And it's very, as far as the graphs go and the data points, these were just right on the money. It was spot on. It shows that cell proliferation stopped, and apoptosis was actually happening in the. So the results seem to be reliable considering they have activity presence. So yeah, it seems reliable considering they have had an effect on cell proliferation. Based on these results, we can say that these two compounds stop cell proliferation and induce apoptosis. The control samples media by itself and the samples containing only cells showed no to little activity of caspase of the caspase activity, which indicates that no apoptosis was taking place, as it should be, because as you can see, the blue line indicates that it was the sample just containing those cancer cell line, and those are just proliferating, so it's not showing any kind of caspase activity because it's not present, and it's, the cells aren't going through apoptosis. So it's a very spot on data as far as our graph goes. I was a little bit confused because I am not 100% sure why our media sample is actually coming up a little bit higher than our cells. That actually to me should be a lot closer than that, but either way it showed very minimal activity, and there shouldn't be any activity present in that. This bar graph is illustrating the amount of caspase activity at 150 minutes in each sample being tested. And we saw this earlier in the previous graph. This is just another graph to just show you the amount of activity that was going on in regards to the caspase activity in each of these samples. Obviously, colchicine and indubulin were those two compounds that were actually having some type of apoptosis effect on those cell lines, and indubulin had a much higher activity, and we were just able to take those points at 150 and plot them right here in this graph and be able to come up with another bar graph to illustrate the amount of activity that was going on at that certain time for the analysis. The second objective of this experiment was actually to determine if colchicine or indubulin causes the caspase activity activated DNase, or CAD, to break down DNA into fragments. The presence of DNA fragmentation is also another apoptotic marker, which can be visualized through a gel electrophoresis. From what we can see is that indubulin shows a nice streak on the gel. As we can see right here, you can see that DNA fragmentation took place right here. And this is just noticeable because when the DNA is fragmented, it gets chopped up into little pieces, which makes it a lot easier to move around in this gel. It's kind of like a size exclusion. The bigger the size is, the less likely it is to move down the gel. It's going to have a harder time moving through the gel. So these smaller pieces can actually move from one side to the other because of that polar effect that the gel electrophoresis has and the charges that the DNA has. So right here's the streak that can be clearly seen in Indubulin. What was really questionable about our assay right here, and where we think, or where I think that we made a mistake, is that during the procedure and prepping for the DNA fragmentation, I do believe we may have switched our colchicine samples along with our cell samples, because in our cell samples, DNA fragmentation is not occurring. Apoptosis is not happening. Caspase activity is not present, which means the DNA is not being fragmented or cut or chopped into pieces. And yet, our well that contains only the cells shows a similar streak to Indubulin. And this is just indicating that there is DNA present that is actually moving down the gel, which was very questionable because it's not supposed to show that. And then vice versa, in colchicine, you can see the DNA band here, right? But that's it. There's nothing else moving down the gel, and so the results have been mixed. So this is where I think that we made an error during the procedures before we ran the samples in the gel, and we may have mixed our two samples into a different well and got these results. So the last assay is actually it's actually called a tubulin polymerization assay. The objective of this tubulin polymerization assay is to determine if the compounds are tubulin stabilizers or destabilizers. The tubulin polymerization assay can also determine the rate of tubulin polymerization in each sample containing a novel compound. So basically, here we're kind of really digging into where are these compounds affecting cell proliferation. And in that sense, tubulin polymerization, to go a little bit deeper into that, beta tubulin is one of the two heterodimers. There's alpha tubulin and then beta tubulin. And basically, these tubulins are needed to form microtubules, which form or are needed to form the mitotic spindle during prophase and metaphase in mitosis cell cycle. The compounds are actually binding to the beta tubulin, which is causing some type of preventing the formation of crucial components for cell splitting. From the data that we were able to obtain, we know that paclitaxel, which can be seen right here as PX, and the purple line, is actually a compound that stabilizes tubulin and actually increases the rate of polymerization, and can be seen right here with this illustration of our. It has a real a high curve. It's a very fast rate that's happening here, where tubulin is just the cells are splitting a lot faster because these tubulins are forming at a faster rate, causing the formation of all the other components necessary to split the cell. The cells and Indubulin, which are right in the middle, actually showed similar rates of tubulin formation, which was kind of questionable, at least for Indubulin, because Indubulin was supposed to have an effect on tubulin polymerization. It was actually supposed to slow it down, but we can see that actually is happening it's actually forming at a faster rate than normal cells, the normal cancer cells. So there may be a mistake right there. I'm not 100% sure what could have happened here, or maybe Indubulin is actually not having that kind of effect on the cancer cell line, as far as maybe it's not attacking or targeting beta tubulin in that kind of aspect. So colchicine, on the other hand, showed a very destabilizing effect. As you can see right here in this green line, that is colchicine right here. And colchicine had a direct effect on tubulin polymerization. Basically, the colchicine sample was the only compound that showed no tubulin polymerization. The tubulin polymerization reaction rate was highest on the sample containing paclitaxel. The sample containing cells only in the indubulin samples has similar rate of reaction of tubulin polymerization. The colchicine was the lowest rate of reaction in tubulin polymerization, which indicates that colchicine was the most effective sample in stopping cell proliferation by binding to beta tubulin in the cancer cell line U937. With that being said, that draws us up to our discussion and conclusion. The compounds that have been tested for their anti-cancer activity, they were tested in regards to through the MTT, IC50, caspase, DNA fragmentation, and tubulin polymerization assays. The data produced from the previous procedures have shown that colchicine and indubulin have had a direct effect on cell proliferation and have been inducing apoptosis in the cancer cell line U937. So with that being said, in the future, I believe we can use more compounds just to screen them, see what kind of effects they have on cancer cell lines. And again, this is just to find, it's in hopes to find a new compound that could eventually become a anti-cancer drug, as another alternative or maybe a better solution to fighting cancer instead of such our normal procedures such as chemotherapy. So just to give some acknowledgments, I really want to thank Dr. Sadana, my lab partners, Tyrone Miner, and Melvin Onya for working with me, and everybody in the cell biology lab that actually contributed by working on different compounds and were able to provide really good data. The end.